Hello, everybody. This is Julian speaking. Let's wait for another 2 minutes since people are still joining the webinar, and then we are ready to get started. Okay, great. I believe we can start now. Welcome everybody to this webinar. My name is Julian. I'm the CSO of Cytina, and with me I also have some colleagues connected to the webinar that help me later with the Q&A session. I'm super excited to host the second webinar of our Selling Cytina webinar series. In April last week, we talked about setting up efficient cell line development workflows. This is something Cytina's technology is well known for. And this week, I will talk about the isolation of bacteria from complex gut samples. So thank you very much for joining. The presentation will last around 25 to 30 minutes. And afterwards, we will have some time to address your questions. For those of you who use the GoToWebinar plugin for the first time, let me briefly show you what you can do here, how to use it. So there's an arrow where you can expand to access further control options. Please note that everybody in the audience is muted. So you, however, are able to submit questions using the text box down here. So feel free and submit questions throughout the presentation whenever they come up, and we are happy to address them after I went through the slides. In case we are not able to address your questions, they are not lost. We will get in touch with you if you want to after the webinar. We also uploaded some additional material in the handout section, like our brochure or app notes that you can download during the webinar. So let me briefly introduce who we are and what we are doing as a company. Since last year, Cytina is part of the Selling Corporation, a young company that has grown rapidly over the last years. Founded in 2016, Selling is now present in over 60 countries, and we have offices all around the globe to develop new products and support our customers. Our product portfolio covers a number of different areas within the life sciences for bioprinting, liquid handling, single cell isolation and analysis, as well as imaging. Here at Cytina, we develop instruments for automated isolation of single living cells since 2014. All our instruments are based on label-free cell isolation and detection and come with image-based assurance of clonality. It started all off with a single-cell printer that you can see here, which was launched in 2015. And last year, in 2018, sorry, we launched the X-site series, which comprises of the C-site and the F-site, two instruments for the isolation of mammalian cells, and the B-site, an instrument for isolating microbial cells. If you look at the largest pharma companies, most of them are already using our technology to generate cell lines or analyze cells. And in addition, we have provided a number of instruments to a lot of well-known labs in academia. This slide provides an overview of the applications that are addressed with our instruments. Our technology is very established in cell line development, which typically refers to genetic engineering of a mammalian host cell line in order to produce therapeutic proteins. For smaller products such as plasmids, large peptides, or simpler proteins, microbial expression systems are often the right choice. The workflow here is somewhat similar to mammalian cell line development, and today we will discuss how our single-cell dispensing technology can be used to efficiently generate also microbial expression strains. Another very exciting application is cultivation of single bacteria obtained from mixed microbial samples. Here, cells often need to be isolated from highly complex microbial samples to derive pure cultures for further identification, for example, for the study of new species from gut microbiota. That's the topic of today's webinar. And the third application is single-cell analysis, and in particular, single-cell genome and transcriptome sequencing. Here we now offer a full platform solution for plate-based assays. Next week, we will host a webinar on single-cell RNA-seq and NGS library prep. So what can you expect from today's webinar? Let me briefly go through the agenda. First, I will spend a few minutes to explain how our single-cell dispensing technology works, for those of you who haven't got in touch with it. Then I will show you how one can use the technology to isolate microorganisms in a label-free manner. And finally, I will discuss a high-throughput workflow for the isolation and cultivation of bacteria from gut microbiota, the topic of today's webinar. The device you can see here is one of the devices of the Excite series, which are benchtop instruments for single-cell dispensing. And if we zoom into the printhead here, you can see the nozzle of our disposable dispensing cartridge. We use an optical detection system, which is somewhat similar to a bright field microscope to detect cells inside the nozzle of our cartridges. And only those droplets that contain a single cell with the right properties are then deposited onto the target substrate. Here you also see one of our cartridges. Before you start the single cell dispensing, the sample is simply pipetted into the reservoir of the cartridge, which can hold anything between 5 and 80 microliter. Now I want to show you a quick video of how this looks like in real time. So here you see one of the instruments in our Excite series, the dispenser. When you start your run, you need to pipette your sample into a new cartridge, which is then attached to the dispenser with 2 screws. And there's also a pipette tip, which is especially useful for mammalian cell samples to prevent the cells from sedimenting in the reservoir. And then you insert a well plate, which is typically pre-filled with culture media or a lysis buffer if it's a PCR plate, and then you can start the run, and this dispenser will go from well to well, dispense a single droplet with a single cell, and after a few minutes you end up with your well plate filled with single cells or single bacteria. All right, let's go back to the slides. I hope you can see the slides again. Next, I want to show you a few slides to explain our technology in a bit more detail. So how do we generate droplets? In this schematic, you can see a side view of our dispensing chip, which consists of a microstructured silicon and a glass cover, and inside you have the liquid with the single cells. This here is the inlet and this is the nozzle. We also have a piston that's driven by a piezo actuator and the microscopic camera system to monitor the cells inside the dispensing chip. During droplet ejection, the piston moves inwards and displaces the liquid in the chip up and downwards. Thereby, a droplet is formed on the nozzle, which eventually breaks up, meaning there is a net flow downwards, which also causes the next cell to move closer to the nozzle. When the piston retracts, new liquid is drawn into the chip. Due to surface tension, the meniscus at the nozzle remains stable, and we are ready to dispense the next droplet. In contrast to, for example, flow cytometry, where droplet generation is based on continuous breakup, we use a drop-on-demand dispenser that's similar to the principle used in an inkjet printer. This results in very low shear forces to not impact the cell viability and enables us to image and sort the cells. How do we make sure that we only dispense the droplets that contain single cells? We now switch the wheel to the front side of our dispenser. This is again a schematic of the nozzle of our dispensing cartridges. You see here the nozzle with some cells inside, and we also have a small pneumatic shutter which is placed directly underneath the nozzle. Using our microscopic detection system and automated image processing, we continuously count the number of cells and other objects in the nozzle of the dispensing chip. If there's no cells inside, the shutter remains switched on and the empty droplet will be removed directly after it has been ejected. In case there is a single cell with the right properties, we switch off the shutter and the cell can be dispensed to the target substrate. If there are multiple cells or cells that do not match the specified properties, the droplets are also removed. I will now show you a video of the software to give you some insight how this actually looks like and how easy it is to set up and start a dispensing run. Please note that this video will show mammalian cells since it's quite hard to share an experiment with bacteria over this webinar since they're so small and difficult to see. But we will look at some images afterwards. So you start with selecting your plate format. In this case, it's a 96 well plate. Which then brings you to a screen where you can lay out your experiment. You can basically select the number, the wells that you want to address with cells. You can choose the cell size and roundness in this case of the cells that you wanna sort and dispense, and also the number of cells. So in this case, we have a few wells, for example, A1, where we dispense multiple cells. In this case, 5. As a control, and then we have a lot of wells with where we want to dispense single cells. The software lets us then confirm that we prepped everything, and then the printing run starts. In the upper right corner, we always have a live video of the nozzle of the dispenser, and you can now see how the cells approach the nozzle and are selected by the algorithm, and the progress can be seen in the well plate here. Each time the check mark occurs, a cell was dispensed into the respective well. There's also a dot plot on the lower right corner that allows you to look at how your sample looks like, how large the cells are in the sample, and allows you to better choose the right gating parameters. It's especially useful for complex samples where you don't know what the cell concentration is or how the cells look like. So I already mentioned that in the Excite series, we have 3 instruments. The C side for mammalian cell detection and isolation, the F side, which comes with an additional fluorescent laser for sorting cells based on fluorescent intensity, and the B side, which comes with a higher resolution optics for the detection and isolation of smaller cells. Let me now show you how this looks like for the B side. For each single cell that is dispensed, a series of images is stored to the hard drive to document the cell isolation process. And this is a compilation of these image series from 3 different experiments. The first one with Bacillus subtilis, and you can see here I marked the target cell with a white arrow, how it gets closer to the nozzle area. Here in the third image, it's detected, and the final image shows the empty nozzle after the cell has been dispensed. And these images can be unambiguously linked to the well that was addressed with the cell. There's also an image series for the dispensing event of a E. coli cell and of an Enterococcus faecalis cell. Let me zoom in a little bit. In this case, it's the image series for Bacillus subtilis. To make this a bit clearer, I will now go through the images, and you can very well see how the cell enters the detection area, marked with a gray circle here, and then leaves the nozzle of the dispensing chip. And here on top, you see the new cells coming closer to the nozzle. They will be dispensed in a bit. There's a growing list of microorganisms that have been isolated with the B side, which really illustrates that the instrument is compatible with a variety of different species and samples. The cell types range from common strains such as Bacillus subtilis and Enterococcus faecalis or E. coli, over Staph aureus, cyanobacteria, and also yeast cells. Since the detection is entirely label-free, the B side is also a great tool to isolate organisms from highly complex samples, such as diverse microbiota. The instrument was applied to gut microbiota from human and mice, but also to environmental samples such as wastewater and sediment samples, as well as clinical samples such as a sputum sample. All instruments in the Excite series use sterilized disposable cartridges to prevent cross-contamination, and they come in packs of 10 and are individually bagged. For the B side, we use a cartridge with a nozzle size of 20 micrometers. For larger cells, such as common mammalian cells, we offer cartridges with a nozzle size of 40 micrometers. Since this is the only part that gets into contact with the sample, there are no routine cleaning procedures required. And this is especially useful when working with pathogenic organisms or clinical samples. It's also important to note that compared to, for example, flow cytometry, there's no risk from aerosols generated by the system, since we dispense droplets in a very controlled manner. Due to the small footprint, the instrument can be placed into regular flow benches. There are no tubings or fluids that need to be connected or deoxygenized. This makes the integration into anaerobic workflows straightforward. Now I want to go through some applications that can be addressed with the B side. Before I talk about the workflow for microbiome research, I want to briefly show you 2 slides on 2 other applications: the generation of microbial expression systems and single-cell genomics. Cloning of bacteria and yeast is somewhat a standard method in the repertoire of every micro or molecular biologist, and there's a lot of applications where cloning is required. For example, in synthetic biology, for antibody fragment screening, or expression of other small proteins, such as antigens, for example. Generation of such expression systems usually start with a microbial host, such as E. coli, Bacillus subtilis, or a yeast strain. And after introducing a plasmid with the target sequence or using other gene editing methods, such as, for example, CRISPR, the cells need to be cloned. And today, this is typically still done by streaking the cells on agar. After streaking, the agar plates need to be incubated to culture the cells on agar, followed by colony picking to bring the clones back to liquid culture. Finally, the plates can be then screened for cultures that stably express the target molecule. This traditional workflow requires several manual steps, and the B site can be utilized to directly clone single bacteria or yeast into liquid culture, thereby bypassing the additional cultivation on agar. And this is not only faster and automated, this also ensures that the cultures are indeed generated from single cells. Another very exciting application is single cell sequencing of bacteria. For this application, the B site allows for precise deposition of individual bacteria into, for example, 96 or 384 PCR well plates. And using low volume liquid handling, individual cells can be then lysed in sub-microliter volumes for subsequent genome amplification. And by combining the B site with our IDOT low volume dispenser, we provide a flexible solution for plate-based single-cell genomics and NGS library preparation workflows. The IDOT is a great tool that allows rapid and highly multiplex dispensing of reagents for cell lysis, genome amplification, barcoding, and this results in an entirely contact-free workflow. After single-cell whole genome amplification, individual bacteria can, for example, sequence one by one. To give you one perspective here, on the right, you can see a phylogenetic tree that we reconstructed from 16S rRNA sequences obtained from single cells isolated from a sputum sample with our technology. We'll talk a bit more about this in the next webinar next week. Now, in the second part of this webinar, I want to guide you through a high throughput cultivation workflow for the isolation of anaerobic gut bacteria that was established in the group of Thomas Clavel at the University Clinic in Aachen. Here we see an overview of the entire workflow that was established to isolate and culture anaerobic bacteria from gut microbiota. It starts with a complex microbial sample that is brought, that needs to be brought into liquid culture. In their workflow, the samples are then pre-cultured for up to 24 hours, or sometimes they are directly processed with the B site. After single cell isolation, the samples are then incubated under anaerobic conditions in the very same flow bench at 37 degrees for up to 7 days, either in liquid culture or on agar. And the first round of identification is then performed with multi-mass spectrometry. The motivation here really is to isolate, culture, and study so far uncultured bacterial species from these diverse microbiota. Such species can be then used to either assemble synthetic microbiota. They might serve as potential candidates for live therapeutics, or can potentially provide new bacterial genes and functions that can be used for other applications in biotechnology. To see whether single bacteria can be isolated and cultured under anaerobic conditions, they first started to validate their workflow with two pure cultures, E. coli and Bacillus vulgatus, the latter one a strict anaerobe. These cells were dispensed into 2096 well plates pre-filled with liquid culture medium. The graph on the right shows results from OD600 absorbance measurements to monitor the growth in the well plates after a day of incubation and cultivation. As you can see, a high fraction of the wells addressed with single cells resulted in growth, 79% for E. coli and 68% for Bacillus vulgatus. Next, 2 complex samples extracted from mice were processed using the workflow outlined before. For each sample, cells were dispensed both into liquid medium and onto solid agar pre-filled into 96 well plates. In total, 25 plates were processed, resulting in over 1,600 wells with growing colonies after 7 days of cultivation. And as you can see here in the graph, more than 80% of the wells addressed with single cells resulted in growing colonies from the complex samples, which was quite encouraging. Out of these, approximately 1,000 samples were used for identification by multi-MS. The resulting bacterial compositions can be seen in the pie charts here. Note that both samples came from enrichment cultures, which resulted in a slightly different relative abundance of individual species compared to the community present in the mice. The group now applies this workflow for the analysis of a variety of different samples, and for those species that do not match the MALDI database they use, they go on and sequence the 16S RNA for further identification. And this already resulted in several isolates of bacterial species that have not been cultured before. This is one example of 3 previously uncultured strains isolated from mouse and human microbiota. This is very recent data that has been presented during a conference in Leipzig around 8 weeks ago by Friesel and Professor Clavel from the University Clinic in Aachen. And I believe this is super exciting, since such culture-based workflows provide a great tool for finding and describing so far unknown or uncultured species in greater detail. It's important to understand that more than 95 to 99% of all prokaryotic species have not been cultured before. This so far uncultured majority provides a huge pool for potential drug candidates or other bacterial functions, as I mentioned before. We also have an application note describing this workflow, which you can download here in the webinar under the handout section, but also from our website. I'm now almost at the end of today's webinar. Let me briefly summarize the key aspects of the B-side, our instruments for isolating small cells such as yeast, bacteria, or archaea. The B-side is able to detect cells from around 1 micrometer to 10 micrometer in size. We use cartridge with nodal size of 20 micrometers. The detection is based on bright field optics, so there's no label needed for isolating the cells. You can use any flat target that fits the SBS footprint, such as standard 96 or 24 well plates or agar, plates filled with agar. And the throughput you can expect is around 3 to 5 minutes for 96 well plate, and approximately 4 times longer for 384 well plate. Due to the small footprint, the system can be readily integrated into anaerobic workflows. We also provide an automation API to integrate the system with plate handling robots or other automation equipment. So how can we come together? We are happy to set up individual telecons to discuss your application and your workflows. After such a telecon, we typically proceed with an on-site demo to introduce you to the instrument and work with your cells, let you work with your cells to study efficiency and growth or your molecular assay. I assume that many in the audience are still affected by the COVID-19 implications. Therefore, we set up a live demo lab, and to show you our instrument. So if you're interested, now it's a great chance to get an application-specific live demo that you can watch from anywhere, and we are then happy to offer demos around the globe as soon as travel restrictions are loosened again. Please get in touch with us so we can schedule either a live or an on-site demo for you. Now we are at the end of today's presentation. This was the second event of our series. Next week we will have a third webinar about single-cell RNA sequencing and NGS library prep workflows. If this is of interest for you, please sign up or forward this to your colleagues. And now I'm happy to take your question. I hope you enjoyed the presentation. So there's a few questions that came in throughout the webinar. Let me start with this. So the first one is, which sample concentration do you recommend, and are there limitations for the sample buffer? That's a good question. So there's of course an optimal cell concentration that ensures high throughput and high single cell efficiency. And this is something, it's not always easy to determine the number of cells precisely when it comes to bacteria or microorganisms. But for example, for E. coli, we know that we typically want to dilute, for example, an OD 0.2 culture around 100 times. And this equals to a cell concentration of around 1 to 2 million cells per milliliter. So that's something which can be seen as soon as you load the sample to your cartridge, and it's also possible to adjust the concentration if needed. But once you have this in place for your sample, this should stay consistent. And the second part of the question, I think, addresses the type of liquids that can be used for dispensing. There are different liquids that are compatible. We frequently use either PBS or, for example, LB medium, but also other media can be used. There's a lot of different media out there for microbial cells. So if it's very specific, we need to test it. But as long as it's an aqueous medium, we should be able to dispense it, and otherwise it's possible to slightly adapt this in order to improve the dispensing performance. The next question is, what kind of sample preparation is required before single cell isolation, which is somewhat a similar question. So for pure cultures, we would typically just dilute the samples. So for example, in the E. coli culture, day culture needs to be diluted with either PBS or LB medium to the final printing concentration. We recommend to filter these media with a sterile filter to make sure that they are free of particles. For more complex samples, such as, for example, fecal samples, like the one used in the anaerobic workflow here, there's a bit more sample prep needed, which basically comprises centrifugation and filtering to make sure there's no clunks or larger objects in the samples that might clog the nozzle. What is the lower limit of detection regarding the size of bacteria? So I mentioned that the system can detect cells starting from around 1 micrometer. This depends a little bit on the contrast of the cells in the sample, but most bacteria species fall within the specs of the B side. So if you're if you have very small organisms, we're happy to look at whether we can reliably detect and isolate them. Another question is, where can I find more information about the single-cell sequencing workflows and about the IDOT? That's a good question. I didn't address this application in detail today. So next week we have a webinar which focuses on single-cell genomics workflows for the analysis of mammalian cells, where we will discuss the capabilities of the IDOT liquid low volume dispenser in more detail for this specific applications. Otherwise, I'd suggest you get in touch with us and we are happy to help discuss your application. You can also find more information on our websites. Can I process my samples in LB medium? So I guess I kind of addressed this. Yes, we frequently dilute bacterial samples with fresh LB for dispensing, which works quite just as well as with, for example, PBS. There's another question, whether the instrument supports solid plate dispensing. I believe the question is, this addresses whether you can use agar as a substrate. And yes, this is actually possible, as long as the outer dimensions of your substrate, which can be also an adapter that holds round plates, fit into our fit the SBS format, so it has the same dimensions as a standard well plate. And if you remember the high throughput cultivation workflow, when I talked about the isolation and cultivation of the mixed samples, some of them were actually dispensed into plates filled with liquid, and some of them were dispensed into 96 well plates pre-filled with agar. So it's also possible to fill agar into individual wells and then dispense the cells. But it's of course possible to just prep agar plates in one big reservoir or plate to dispense arrays onto this agar plate. Using well plates here has the big advantage that when you look into complex samples where you often find species with different growth rates, then the wells really make sure that your clonal culture is confined and fast growing species will not overgrow slow growers, which can be quite useful. There's a few questions on robotic automation. I already mentioned that we offer an API for our software, so the system can be controlled from a separate computer to integrate it into fully automated workflows. It's possible to use plate handling robots. I guess we need to discuss the details. We need to spend a bit more time to discuss the details here. We can kind of help you choosing the right gripper that's compatible with our instrument. But in general, this is possible, yes. So let me see if there are more questions that I haven't addressed so far. Some are somewhat similar. There's one more question. What's the difference of the cartridges for the B side? So for dispensing bacteria, we use a cartridge with a smaller nozzle size compared to when we work with our instruments for mammalian cell isolation. For the B side, we work with 20 by 20 micrometer cartridges and nozzle sizes. And for mammalian cells, we would typically work with a nozzle size of 40 micrometer. But the principle and how we detect the cells is similar. I think I addressed most of the questions. For those questions that I haven't addressed, we will get back to you after the webinar to discuss this in a bit more detail with a bit more time. I hope you enjoyed today's session, and I wish you a very good rest of the day.